Okay, my name is Arailym, and today I will be presenting synergistic topic on the synergistic removal of pharmaceuticals by polymeric membranes. And here is the name of my co-authors: Damira, Aliya, Professor Jong, Professor Stavros, and Professor Elizabeth Arkhangelsky. Here is the outline of this presentation. I will give brief introduction. Then introduce materials and methods used in the experimental work, results and discussion, followed by conclusion. So water pollution is one of the most challenging environmental problems in today's world. Presence of harmful contaminants of emerging concern, shortly CECs, in water resources are increasing every day due to the rapid development of different industries. However, there is limitations related to the removal of CECs by conventional wastewater treatment plants. These type of pollutants have adverse health and environmental effects even at very small concentration. So in order to avoid any kind of health problems, we have to make sure that these kind of contaminants are absolutely removed from the water bodies. What are the CECs? This is the different chemical compounds which are mostly toxic. There are different types, including organic dyes, pharmaceuticals, nanoparticles, personal care products, cyanotoxins, and pesticides. These type of materials are or chemicals are not regulated by law and could pose to the harm to the environment and human health. There is no national or international regulations dedicated to the controlling and monitoring of the CECs. They could enter and accumulate inside the living organisms and are very dangerous. Pharmaceuticals are constantly consumed by humans, or the animals can enter the environment via different pathways. Next. Pharmaceuticals. The improper disposal of pharmaceutical waste by household and hospitals can lead to the release of the uncontrolled amount of the pollutants into the water and soil systems. The veterinary pharmaceuticals can enter the soil medium or the surface water bodies through the animal excretion of urine or the feces. Pharmaceuticals are persistent and not easily degraded by naturally. So as a result, many of them cannot be fully removed by the conventional wastewater treatment plants. And our recent research studies demonstrated that in Kazakhstan, wastewater treatment plant, most of the pharmaceuticals are not fully removed. In order to solve the problems, we propose the introduction of the membrane filtration. Membrane separation technique is recognized as one of the most efficient water treatment technologies in today's world. Also, we selected Aspirin, Carbamazepine, and Sulfamethoxazole as the target pollutants because these pharmaceuticals are one of the popular medical drugs and considered as top priority medical substances in many different countries. Sulfamethoxazole is an antibiotic. It's not easily biodegradable and challenging to break down. It has a high persistence characteristics. Carbamazepine is an anticonvulsant medication. It's also similar to sulfamethoxazole, has a persistent nature and has low water solubility. Aspirin is an analgesic, water soluble, and compared to the remaining, it's biodegradable compound, but it's one of the most commonly detected in water bodies and wastewater treatment plant effluents in many different countries. Methods and materials. We selected two types of polymeric membranes for the filtration experiments. First is polycarbonate track H membrane with 10 nanometer pore size. Second is polyethersulfone polymeric membrane with 200 kilodalton molecular weight cutoff. And the contaminants selected for the feed solutions was of an analytical grade: Aspirin, sulfamethoxazole, and carbamazepine. The initial concentration of drugs used in the feed solutions were 3 ppm or 3 milligram in 1 liter feed solution. The removal of the pharmaceuticals by the membrane is calculated by this equation. It's simple feed per meter over concentration feed and by the percentage. Next. In the figure 1, you can see the lab scale filtration setup. The feed tank will be placed with the feed solution on the stirrer plate to ensure that the feed solution is homogeneous throughout the filtration period. The feed is supplied by the peristaltic pump in the dead end flow to the membrane cell by the active area of the membrane facing the feed, and then the permit samples are collected in the permit tank, placed on the digital balance, and the numbers of values of the digital balance is automatically collected every minute to the computer. Next, equipment. The concentration in the feed and the permit solutions are analyzed with the HPLC system with the C8 column, and elements used to the mobile phase is ultra pure water and acetonitril. Also, samples are filtered with the 0.2 micrometer filters in order to make sure the accurate analysis of the feed and the permit concentration of the drugs. The zeta potential of the polymeric membranes is analyzed by the surface electrokinetic analyzer, and the contact angle of the membranes is measured by the OCA 25 by standard sessile drop protocol. Next, the result. You can see from the figure 2 the removal of aspirin by both membranes. The blue bars are results for the PST membrane, and the red are results for the PES membrane, removal of the aspirin. You can see that overall the blue bars are higher, indicating that the PST membrane overall demonstrate the higher removal values compared to the PES. The absolute elimination, like 100% removal of the aspirin drug, was achieved with the aspirin with sulfamethoxazole combination for the both polymeric membranes, 100% for each. Filtration of individual aspirin in ultra pure water demonstrated excellent results, also 97.15 and 89.98 drug aspirin removal values. The lowest aspirin was in the aspirin carbamazepine drug combination. You can see by the red, the lowest was 3.23% removal, which is very low, indicating that all of the aspirin remained in the feed solution. The average removal results by the PSTA membrane 75.12, by the PES membrane 15.3 points. Next, on the figure 3, there is the results for the carbamazepine removal. The individual pharmaceutical removal indicates that PSTA 61% and the PES 17%. In this carbamazepine removal, also PST membrane demonstrated higher removal values of the carbamazepine. Next, the results of the sulfamethoxazole removal by both membranes. In this case, there was very conflicted results with the aspirin combination, the absolute removal by the PST membrane and the zero removal by the PES membrane. The average removal values, again, PST membrane demonstrated higher results. And the hydrophilicity of the membranes was assessed by the contact angle of the polymeric membranes. The higher angle indicates more hydrophobic nature. PST membrane 75-degree contact angle and the PES membrane 90-degree. And for the studied drugs, the carbamazepine was the most hydrophobic, followed by aspirin, and sulfamethoxazole was relatively hydrophilic. Comparing the hydrophobicity results, we can conclude that hydrophilic filter demonstrated higher retention values, and the presence of the most hydrophilic drug SMX resulted in the lower removal values in the majority of experiments. This can be due to the stronger attraction between the hydrophobic drugs to the porous materials made of polymer. Next, membrane charge as function of pH was studied by the zeta potential. However, the zeta potential of the both polymeric membranes was similar. It was between the -20 and to -30 millivolts. And also it was around -25 millivolt at neutral pH. And for the medical drug charges, aspirin and SMX are negatively charged, while carbamazepine is neutrally charged. So negative drugs are possibly interacts with the neutral drug, and this further influences the removal of the drugs. The average retention values of the aspirin, carbamazepine, and SMX are concluded in this text, like aspirin, carbamazepine, SMX. The highest was for the aspirin and the lowest for the SMX. Each experiment performed demonstrated its unique rejection values. So this could be due to the removal of the drugs was affected by the presence of the secondary or third pharmaceuticals in the feed solution. Polycarbonate track H membrane demonstrated higher removal values than the polyether sulfone membranes for all three pharmaceuticals. The average retention values are the following order: highest aspirin, followed by carbamazepine, and then final SMX. It's important to note that all the pollutants in the study have molecular weight less than the pore size of the pore size of the both membranes. So we can say that the pollutant removal did not happen only due to the size exclusion. Assuming the interaction between drugs and simply summoning their molecular weights, the calculated values still do not exceed 10 nanometer or 200 kilodaltons. So here is the conclusion of this research study. Synergistic removal equal to 100% of aspirin and SMX happened when both pharmaceuticals are present in the feed, aspirin and the sulfamethoxazole combination. The presence of the second and third medicine in the feed could improve or reduce retention capability of the membranes. All three mechanisms of the pollutant removal, which are size exclusion, charge, hydrophobic interactions, are involved in the removal of studied pharmaceuticals by PES and PST membranes. There is possible interactions between membrane and drugs, and while pharmaceuticals require further investigation, we can conclude that polymeric membrane filtration can be a possible solution for the pharmaceutical removal in the wastewater treatment plants. So here is the reference. Thank you. And here is the references used in the study. If you have any questions.
Thank you for your